Now we have Dr. Ledesma-Amaro. Give me 1 second. I will share my screen. We have Dr. Rodrigo Ledesma-Amaro. He's a reader in synthetic biology of the Department of Bioengineering in Imperial College London. His research focuses on the development of new synthetic biology tools that allow to precisely manipulate microbial cells in a reliable, predictable, and standardized way. In particular, his group is interested in cutting-edge techniques that permit a fine-tuning of different metabolic pathways for the production of chemicals, fuels, diverse materials, and among other applications. Today, Dr. Ledesma is going to talk about his title. His talk is Synthetic Biology and Engineer Microbial Communities for Sustainable Bioproduction, which is part of the PsychoLim project from the ERACO Biotech. Dr. Ledesma, thank you very much for joining us today. I'm just going to stop sharing my screen for a second. Stop sharing.
Thank you very much. Thank you for the invitation to present our project. I'm going to share my screen now.
Yes.
You should be able to see my screen.
Yes, now, yeah.
Perfect. So I'm going to, instead of choosing to tell you the details about one particular project, I'm going to talk about a little bit of high-level projects that kind of emerged from this PsychoLim project and the concept of microbial communities. And starting by introducing the people who are behind the pipettes and been doing the work. So this is my group, and we are based in South Kensington, in the museum area of London, if you've been here. And we are based in this South Kensington campus. So my main research is about sustainability. And this PsychoLim project is also around sustainability, as most of the ERA biotech projects in this case. So we're also interested in kind of contributing to the sustainability by bioproduction, as the talk that we just had, and using microorganisms as well. So microorganisms, as you know, have a diverse variety of metabolism with thousands of molecules that can be produced. And some of these molecules have different applications, from biofuels, chemicals, foods, pharmaceuticals, etc. They normally are produced in small amounts, and we therefore need to engineer these organisms to produce them in higher amounts. So we can achieve the affordability that allows us to scale up the processes. So when we define a bioprocess, we can choose many different hosts, and we work with a variety of hosts, yeast and bacteria. My group is specialized in yeast, and the PsychoLim project we're combining both yeast and bacteria. So these are different organisms we are working with at the moment, and the different size of the organism in the screen is proportional to the number of people in my group working with that organism. So as you can see, we mainly work in yeast, and our main yeast are Yerovia politica and Saccharomyces cerevisiae. So we like this idea of engineering communities, and I'm going to have the second part of my talk focus on that. But before getting to that, which will be one of the research lines of my group, this is microbial communities, and this is the core of the PsychoLim project. I'm going to tell you how we can get to engineering communities by the other two research lines that we have in my group. One is the developing of synthetic biology tools that allow us to manipulate pathways in a more efficient way, and then bioproduction, so manipulating those pathways using these tools to maximize the production of specific molecules of interest. So one of the tools that we've been developing and we use quite a lot in the lab is CRISPR. So I think by now most of you have heard of CRISPR. This is the molecular scissors that allow us to edit the DNA in much more efficient way than we were able until a few years ago. But CRISPR has also the ability to achieve multiplexing. That means we can manipulate multiple targets in the genome simultaneously. So that means that the way CRISPR works, you have the Cas enzyme and guide RNA, the Cas enzyme bind the guide RNA, and the guide RNA is going to be complementary to a specific region in the genome that we can define when we design our guide RNA. Therefore, the Cas is going to bind the genome in that particular region that the guide RNA guide, and then make the effect. And the effect can be deletion, it can be making a cut, it can be overexpressing a gene by fusing on transcription activator, or it can be repressing a gene by blocking transcription. So in metabolic engineering, when we want to manipulate pathways to maximize a product, we usually want to overexpress some genes, repress some other genes, and we usually go one gene at a time, and with traditional tools, or couple of genes at a time. Like we overexpress one gene, we delete the other gene, and that takes time when you are thinking about changing 10, 20 genes. What we wanted to do with this tool is being able to, in one single transformation, being able to upregulate or downregulate a large number of genes. And we developed this tool to control the expression simultaneously, upregulation and downregulation of up to 24 different genes in one go. So one example of application was for the production of succinic acid, which is a building block for the chemical industry. It has also properties in the food preservation industry and etc. And as you know, this succinic acid, succinate, come from the TCA cycle, and you can link the carbon source or glucose directly to the TCA cycle to produce this. And this is a pathway that has been studied quite a lot in the literature. So we knew already which genes we can downregulate or upregulate. So downregulate are those in red, upregulate are those in green, to really make the conversion of the carbon substrate into succinate. So we developed this guide RNA array, which is a lot of guide RNAs put together in one single cassette with the targets that we wanted. And by qPCR, we could see that downregulation path targets were downregulated and the upregulation one were upregulated. And at the end, we also made the system inducible. So when we add the inducer, the system activates, and we can see the increase in succinic acid compared to the non-induced engineered strain. So we use these and other tools for a variety of different products, and we've been working in also using low-cost substrate, and this is part of the Sickling project, where we were targeting using raw glycerol as a waste product from some industries. But we've been working in mucilaging materials, starch, and others in a variety of different products that can be biofuels or nutraceuticals, pharmaceuticals, etc. So more recently, we've been interested in food and this microbial food revolution. Like, we need more sustainable ways of making our foods and ingredients, and microbial bioproduction is a way to achieve so. And therefore, we decided in the Sickling project, we targeted limonene production, which is a molecule that is highly used in the food industry, not only. So we've been doing metabolic engineering with this tool for making a variety of ingredients, including limonene that I show here, but we're also working in other aromas, preservatives, colorants, sweeteners, antioxidants, lipids, and proteins. And after this, I moved to the real topic of the talk, which is the microbial communities, how we can envision the bioprocess of the future. Is going to be something like that. It's not only one organism engineered to convert feedstock into a product, but a variety of microorganisms that coordinate their behaviors and maximize the conversion of that feedstock into a product by things like recycling waste products or byproducts, or maximizing the utilization of different sugars simultaneously. So for achieving that long-term goal, we need to start understanding and developing tools to engineer how different strains interact with each other. So one way we can design communities is by using division of labors. It is splitting a metabolic pathway between two organisms, reducing the burden of expressing the whole pathway in one single organism, which could affect normal growth. Another mechanism often found in nature is syntrophy or cross-feeding. This is the exchange of essential nutrients from one organism to the other, and this creates robustness in a synthetic community, because then we will not expect that one community will overtake the other, because it still depends on the slower growing one. And we also need to control populations. This is if we want to have more active a specific part of our process than another, we need to be able to control that to the level that we want. So we've been developing tools to achieve that, and I'm going to give you like very high level overview of some of the projects that emerged from the Sicolim project and other related works. So one of the communities that we engineered was to produce raspberry ketone. So this is the aroma that we recognize as raspberry. So the good thing is when we were working on this project, really the bioreactor room were smelling really, really good. Everyone was hungry all the time. And we create these communities by splitting division of labor, splitting the pathways into 2 or 3 organisms, and also by controlling the population ratio by just changing the inoculation ratio. So we could see how different communities behave differently, but some communities produce as much as the non-communities bioprocesses. So another project was in the production of malonic semialdehyde. This is also a chemical precursor for bioplastics and other products. And we also combine division of labor, like splitting a pathway in 2 in a way that the first strain produced the intermediate beta-alanine and the second strain produced the final compound, malonic semialdehyde. In this case, we combine the division of labor with different ratios, but also with cross-feeding. So the third of the 3, if we go back, the third of the mechanisms that we wanted to engineer. And this cross-feeding actually was interesting because there's a link with the talk we just had before, because this is actually a tryptophan intermediates and indole being exchanged between one and the other to maintain these populations in a specific way. And we saw how some microbial communities, these tryptophan cross-feeders communities, produce 2 times more of the final products than the wild type or the engineered non-community. And this one, this work was published actually yesterday, so now you can read it if you want. It's now online, and we also did division of labor in this case for the production of another food ingredient, which is resveratrol, an antioxidant. We split the pathway in the fumaric acid step, so these strains are sharing fumaric acid, and the other one is making the final product. And we combined it with cross-feeding as well. And in the innovation in this work was the way we engineer this cross-feeding to happen, allow us to control the population levels in the final bioprocess. And again, we found that some communities, not all, were able to produce 2 or 3 times more resveratrol than the non-community, non-cross-feeding one. And more in concrete about the Sicolim work, as I said, the initial core project was about limonene, and limonene happened to be a tough molecule to produce, and monoterpenes are toxic to the cells, so we had to do quite a lot of engineering. We have to redirect the mevalonate pathway that produce the precursor and limonene to the peroxisome. So this is an organelle in yeast, and by transferring this pathway into specific organelle allow us to reduce the toxicity, and with this allowed us to also increase very significantly the titers of limonene. And what we are doing as well in collaboration with some of the partners is engineering this division of labor, so limonene can be converted into other products of interest, such as menthol and other aromas. And that's all from my side. Knowledge to all the people in the group, specifically those that work in Sicolim project, and all the funders, particularly Raco Biotech. And thank you all of you for your attention, and I'll be happy to answer any question.
Thank you so much, Rodrigo. That was a great talk. And I have is a very technical question. So when you did the guide RNA array of 24 guides, so to separate them, did you use like a cleavage kind of like a hammerhead tRNA or something like that?
So yeah, it's a good question. I didn't go into the details, but yeah, you need to process the guide RNAs for them to be active, and there are many ways to do that. So you mentioned some of them. The one we use is CSY4, which is another endonuclease that we express separately and recognize a specific cleavage domain. So we introduce this cleavage domain between all of the other guide RNA. And the advantage of this is this cleavage domain is smaller than a tRNA or other, so reduce a little bit the repetition of the array.
Now it's like 24. Wow. So we have a question here in the Q&A. It says, how are you ensuring that the DCAS repression or activation effector does not compete and have different degree of effectivity?
So it's I mean it's a very good point, and the only way to really know the degree of effectivity is testing it. So for example, in this case, we did qPCR to test how well the guide RNA was repressing, and how the number of guide RNAs could affect that repression level. If you don't test it, it's very difficult to predict. What so far we haven't found a limitation of expressing more guides and lower efficiency somehow. We haven't studied also in very detail, but we think there's still capability for Cas to have more guides and still be effective. So I didn't show it, but in this work we also generated a library of strain with different expression level of the Cas enzyme or the guide RNAs, and we can tune. If you need more Cas to guide RNAs, you can potentially use one strain with a higher promoter. In our case, for the 12 and 24 guides, we really are using a very low expression Cas level, so it's not affecting the cell metabolism, and it's still very efficient with the guide RNAs.
And we have a second question here. It says, would sequestering limonene to peroxisomes affect limonene recovery and downstream processing?
So yeah, I think I was a bit unclear with that. So limonene is not really sequestered in the peroxisome. So limonene is going to diffuse to the media, and it's a similar process to the indole that we were seeing just before, when we need a second layer to extract it in the end. It kind of reduced the interaction, if you want, of the diffusion time through the other parts of the metabolism, and somehow that helps a little bit. I think toxicity is not the only issue that we are finding why we have low titers. I think there are other regulation going on, especially in the monoterpene synthesis, that need to be more active, and we're still doing some engineering on that area.
We have another question here. So it says, Cas itself induces a lot of toxicity. How stable your strains are in larger scales?
Yes, a good point. So I mean, in this case, because Cas is not cutting DNA, it's just binding DNA, repressing or activating, it's not as toxic. We haven't seen a lot of toxicity coming from the Cas. What we have seen is toxicity coming from guide RNAs that bind some native genes, as in like, for example, some glycolysis pathways, just because some of the promoters that we use in synthetic biology happen to be also native promoters. And that we learned through the process that because our guide is targeting our synthetic promoter, is also targeting the native pathway sometimes. And that creates some sort of burden in the cell, and the cell can evolve and kind of recombine this guide RNA. So that happened, and we found that was an issue in the paper that we saw. And the way we got around that is by creating this inducible system. So we only induce the expression of the guide RNAs at a certain time in the process. So there is no this evolution pressure, because during the growth phase there is not so much burden.
I have a question. So about the co-cultivation of the different engineered strains. So it's like, so how do you maintain the balance? Is like, do you put the strains together, and that's or what kind of input do you have to give like during the process?
Yeah, that's a good point. So we can see, for example, here, let me see, or here. So yeah, the initial inoculation level is going to change the dynamics of the system. And in the article, we have some growth curves where you can see how different inoculation rations really change when the two populations and so on. But what we found interesting to control the population more controlled, if you want, is the cross-feeding. So here you can see these pathways, they are like cross-feeding pathways. So in the case of tryptophan, it was the one that one need to exchange intermediate of the pathway and tranilate, and the other is going to convert it into indole and tryptophan, and is going to change the tryptophan or indole to the other cell. So that, because it has a specific dynamics of how much one cell secrete one metabolite and how much the other needs and uptake, that creates some dynamics as well, and that control the populations in certain way. So we still don't control fully how this population established, but we have developed kind of a toolbox, and this is what this paper is about. We create a toolbox of strains, each one with different type of dynamics, if you want, so you can go and pick that particular co-culture that is going to have a particular growth dynamic.
When I have before. Yeah, I can't find it. Yeah, I think I don't see more questions in the Q&A. Yeah, that was great presentations. Thank you very much, Dr. Rodrigo. I had another question. I can't find it. Yeah, I remember. So I'm curious about like what is kind of what precautions do you need to have when you work with like an environment like a bioreactor and you're working with genetically modified organisms? Do you need to kind of like it has to have a function of autoclave, as in like sterilization of the full thing after you finish the process, or how does it work?
Yeah, we do follow the normal procedures. We work mainly at lab scale, so we need to autoclave basically kill all the biomass at the end once we finish working with it. So after the bioreactor, you take the samples, you analyze whatever you want, but then we make sure we autoclave all the culture broth, so there is no GMO cells or GMO DNA that can escape.
What is the function of the involvement of the company? You're in your consortium, there was a company involved. Did they put this in a bioreactor already?
Yeah, so they are doing it that now. They've done already some bioreactor, but they still do.
Okay.
They're still doing some. That's why the article is not yet submitted, because we are now trying to scale a little bit and optimize conditions in bioreactor. Yeah, the company is ACS Bio in Slovenia.
Fantastic. Both of the talks. Thank you very much.
Can I just
yes,
mention one thing I wanted to have mentioned before, but just what we are hiring at the moment was postdocs and PhD students. So if anyone in the audience is interested.
Oh yeah, I have been receiving lots of questions from people like, yeah, I want to work. Yeah, so I would just make sure to get the word out that you're looking. So both PhDs and postdocs.
Yeah.
And this is to be in Imperial College.
Yeah, that's right.
Based there. Okay, perfect. I think we have an extra question. Oh, okay. One last question. It says, was the limonin work done with Saccharomyces or Yarrovia?
Yarrovia.
With Yarrovia. Okay, yeah, perfect. I just want to kind of close the webinar here, and I want to thank very much to my speakers, Dr. Catarina and Dr. Rodrigo. Thank you very much for answering all our questions. I want to invite everyone to our next webinar. That's going to be on February 22nd at 10 a.m. And we're going to have Professor Atisber Lopez Cortajarena from CIC Biomagune talking about engineered proteins as scaffold to assemble multi-enzyme systems. So yeah, so this is going to be a very interesting talk. And Professor Alison Smith is going to be talking about developing algal production platforms for high-value chemicals. So we're going to have two very different talks. One is going to be protein engineering, and the other one is going to be algal biorefineries. Again, thank you very much, everyone, for your questions. Thanks to our panelists, and I'll see you in our next webinar. Thank you so much. Bye-bye.
Thanks.
Thank you. Bye, Catarina.